my pleasure today to introduce Jenna Judd, who will be giving her finishing talk. Jenna grew up in the Lake Tahoe area and spent her childhood mountain biking, skiing, and hiking in the Sierras, which I think gave her her initial appreciation for natural history and certainly began to focus her interests in biology. Jenna did her undergraduate work at UC Santa Barbara, where she studied marine biology, but it just wasn't coursework that she excelled at. She also got involved as a researcher with a coastal monitoring program, also looking at biogeographical distributions and sea urchin recruitment. She furthered this research experience when she went with the studying abroad program to Queensland, to Brisbane, University of Queensland, Brisbane, and basically worked in their terrestrial and marine ecology program for a semester. She then served as a course assistant at the Woods Hole embryology course in 2008, where she met Nipam Patel and sort of began her transition or her transit to Berkeley. And it was really two professors at UC Santa Barbara, though, Rachel Heyman and Jim Childress, which sort of got her thinking about the deep sea, and particularly deep sea biodiversity, and how she could study chemosynthetic communities. However, by coming to Berkeley, Jenna would not be going into a deep sea biology program, and she'd have to find an alternative in terms of her deep sea studies. And she went out and did this. First, Jenna applied for the NSF East Asia and Pacific Summer Institute, which she successfully achieved, and ended up going to Japan, where she worked with Takanori Sasaki and Yorosori Kano, two Japanese leading researchers in mollusca in the deep sea, as well as in reducing communities. Jenna then successfully completed an application to the DAAD Award Scholarship, which took her to Europe, where she worked in Munich with Gerhard Hausbrunner, and this provided her entree into the European deep sea and chemosynthetic community group, most notably people like Philippe Boucher and Anders Warren. So this access to the Europeans basically provided access to specimens, techniques, and ultimately we'll hear about today in the second part of Jenna's presentation. However, Jenna still needed to get to the deep sea, and Berkeley does not have a Navy. We're not Scripps, we're not Woods Hole. However, there was an establishment down the coast, the Monterey Bay Aquarium Research Institute, where with contacts through Bob Reinhoek and Jim Berry, Jenna basically was able to design, organize, and logistically pull off a tag-along experiment on one of the cruises that Jim Berry took out into the Monterey Bay Canyon. With this, she basically handled the design, secured the wood, got the logistics of getting it down onto the ship, getting all the specimens back. This is really a difficult thing to do for most scientists, much less a graduate student. And we'll hear about this accomplishment, which not only logistically was difficult, somehow she managed to get back and get all of the bundles pulled up 2 years later after sitting on the bottom in the Monterey Canyon. That was probably the most amazing part, and a certain amount of serendipity, of course. Besides this graduate career, Jenna has continued to have a life filled with travel and research, as well as outreach. She maintains her own blog, The Eclectic Limpet, and has also contributed to the Oakland Unified School District CalBlast program with UC Berkeley, as well as taking on both tours, as well as Cal Day, and being involved with UCMP and IB, for which we're very much appreciative. I think we can all agree that Berkeley did not have a deep sea program when Jenna arrived, but Jenna basically created that program while she was here, and today she'll share the results of that program that she put together here in her talk entitled Patterns of Specialization in the Deep Sea at the Individual, Ecosystem, and Evolutionary Level. Jenna.
Thank you, Dave, for that wonderful introduction. I feel very fortunate and grateful for all the opportunities I've had since I've been here. And so before I get into talking about the different patterns in the deep sea that I've studied, I'm just going to give you some background on the habitats that I've focused on while I've been here. So first, just to show you a picture of the deep sea, this is what it looks like. So it's really dark. It's typically characterized by cold, stable temperatures and high pressures. But, and you might not expect there to be very much life down there, and historically people didn't think there was much life down there. But when we turn the lights on, it actually looks like this. So this is near my field site south of Monterey Canyon at around 3,000 meters. And you can see it's far from devoid of life, and the deep sea is actually one of the most biodiverse places on the planet. However, it is highly limited by low productivity levels. So most of the animals are relying from food sinking from the surface, and most food is recycled in the shallow layers of the ocean. So most of the deep sea is characterized by low biomass and low metabolisms. We see the opposite of this, however, at places like hydrothermal vents. So you can see here this very high biomass of these specialized tube worms called Riftia, which actually have some of the highest growth rates of any animal on the planet. And they're also dealing with highly fluctuating temperatures and high levels of toxic compounds like sulfide. So to deal with this, but they're there for a good reason. So most of the ecosystems on the planet are relying on photosynthesis, as I'm sure you know. Energy from the sun is fueling chloroplasts to fix CO2 and organic carbon, which is fueling most of the life we can observe on Earth. And a different process of primary production is found at hydrothermal vents, and this is called chemosynthesis. So rather than sunlight fueling this reaction, it's reduced molecules like sulfide, and in other cases there might be methane or other compounds that can fuel these microbial metabolisms. So it's the microbes who are doing the work and fueling these ecosystems. But it's not just hydrothermal vents. We also see similar ecosystems that are fueled by chemosynthesis at cold seeps, which are found along continental margins and other places where subduction might be occurring. And I forgot to mention that hydrothermal vents are most commonly found at mid-ocean spreading centers where plates are moving apart. And then we also have chemosynthesis fueling ecosystems at organic substrates such as whale falls and wood falls. And so all of these ecosystems are linked together because they have the potential to fuel chemosynthesis, and they are typically lower in oxygen and have some level of sulfide or methane or fuel for that metabolism. And they also, as they've been explored, so hydrothermal vents were discovered in 1977, and since then there's been more exploration of these habitats and better technology for observing them and sampling the organisms that live there. And researchers have observed that there are links between these communities, not only in the way they're obtaining their energy, but at taxonomic levels as well. But of course, there are definitely distinct specialists at each of these habitats, including some animals that can consume bone directly and wood directly, which are obviously not living at vents or seeps. So throughout this talk, I'm going to be referring to reducing habitats, and by this I mean any habitat that has that potential to support chemosynthesis by providing reduced molecules like sulfide or methane. And this typically means hydrothermal vents, cold seeps, whale falls, and wood falls, but I will also be talking about some other organic substrates that are tied to these habitats. So to get into the main questions of my dissertation, I've focused on whether there's a common evolutionary path towards specialization in reducing habitats. I'm interested in the different ways animals have diversified when given the opportunity. And then once lineages have become specialists in these habitats, how have they modified their anatomy to adapt to and fit into that lifestyle? And what can we learn from looking at their anatomy when we can't study them directly in these difficult to access environments? And then lastly, from the perspective of the habitat, how does variation in sunken wood influence community assembly? So seeing the importance of the environment shaping that diversity. All right, so as I mentioned before, since the discovery of hydrothermal vents in those rich communities, subsequent research has attempted to link together these different habitats through ecological and evolutionary processes. And one of the most famous hypotheses is the stepping stone hypothesis proposed by Craig Smith in 1989. And since then, there's been a lot of research testing the potential for animals to make it in an ecological timescale between these different habitats, whether certain lineages we can trace their evolutionary trajectory between these, and whether there's any kind of pattern at all. And so there's kind of a mixed bag of patterns, though, and they've actually addressed the stepping stone hypothesis in a recent review this year, if you're interested in reading up on it, and found that different lineages have utilized these habitats in a range of ways to get to their current distributions. But I was still interested in examining this early on in my graduate career, and wanted to look at whether we can see consistent patterns over a large, diverse group. And so the group I chose was the gastropods, because they're actually one of the most diverse groups occupying these reducing habitats. And they're occupying them in a range of ways, from limpets that are grazing biofilm and bacterial mats, and these guys are also grazers, to opportunistic predators and scavengers, to those that have highly modified morphologies to deal with the conditions at hydrothermal vents and accommodate symbionts. So this animal has a large gill and a reduced stomach because it's mostly relying on fuel from the symbionts living on its gill. And then those that can consume bone directly, the ruby spira, or wood directly, this pectinodontid limpet. So as I began gathering information on each of these groups and where they lived, whether they lived on vents or seeps or wood or whale falls, I realized that through time, as more discoveries have occurred, there's been a tendency for some groups to find expansion in their ranges. So groups that were once deemed to be specialist in one habitat or another, we've subsequently discovered that they're actually a little bit more flexible than we originally thought. So, and this is due to the deep sea being highly undersampled, and so I feared that there might be a potential for a false signal of endemicity if I just coded them according to what habitats we knew them from. So to make my framework a little bit more flexible, I coded each gastropod genus as occupying no reducing habitat, so it's never been found in any habitat, any of the 4 habitats I focused on. Whether it's in a mix of non-reducing and reducing habitats, so these would be generally the scavengers or opportunists that can be there if they want to. Those that are in multiple reducing habitats, so maybe vents and seeps, and those that are in just a single reducing habitat. And my questions were to test whether lineages followed this kind of straightforward trajectory from generalist to specialist, whether different clades have different patterns or might get stuck at one state or another, and whether there's any kind of pattern at all. So to do this, I wanted to use a phylogenetic context, of course, but there weren't any available trees that covered the span of all the gastropods and included enough taxonomic resolution to be able to map these taxa. So the solution was to build a supertree. So this was work that I did with my advisor Dave and Lucy Chang in the Marshall Lab. And so the way this method works is to take many published trees that have overlapping taxa, and then by running these algorithms on the topology to find agreeing relationships, you end up with a resulting much larger, denser tree through which then we can use to ask ecological questions or character evolution questions. And so this was a big challenge in itself because of the available gastropod phylogenies that are out there. And I'm not going to go into all those challenges today, but the important thing is that we arrived at a solution with 462 gastropod genera. And I don't expect you to read any of those labels, but starting at the base, we have the patelogastropods here in yellow, then the vitigastropods in blue, Neomphalina in red, Neridomorpha in pink, Heterobranchia in orange, and then this large green clade is the cenogastropods. And so the main pattern that we observe when looking at this tree is that most of the branches and most of the tips are white. And so this indicates a pattern of no chemosynthetic occupation, and this makes a lot of sense. And it's generally, lineages have infrequently invaded these habitats, and when they do, they've typically been one-off expansions into those environments that have rarely resulted in a major diversification. However, an exception to that is the Neomphalina, which is 100% coded in reducing habitats, and most of these genera are vent specialists. However, some of them live in multiple habitat types. And this branch is a little bit contentious, and it could actually be embedded at the base of VD gastropoda, but this is sort of the state of the knowledge given the available phylogenies that we could put in our tree. So that will stay as it is for now. And the VD gastropoda are also exceptional in that they have the most lineages out of all the clades that have invaded these habitats, and also represent several unique specialization events. On the other end of the spectrum, we have the Gastropods, which are our largest and most derived clade within Gastropoda, and they, for the most part, have these one-off green lineages which indicate that they live in a mix of reducing and non-reducing habitats. So a lot of these are general deep sea taxa that also have the ability to act as scavengers or predators, and usually at the periphery of these environments. But the one exception to that is this clade down here called the Abyssochryssoidea, and they represent a lineage that has highly modified its anatomy to specialize in these environments, and it includes this vent snail Elviniconcha that has that enlarged gill for housing symbionts, and the ruby spira, which is the only known gastropod to feed on bone directly. So if we want to look at the patterns in number format instead, the main takeaway is just that different clades have different numbers of lineages and proportions of those clades occupying these habitats. And also the main pattern is that of all of the lineages that are coded in these habitats, there's a nearly equal number that are in either a mix of habitats, so both reducing and non-reducing, or only in a single one. And there's a lower number that are found in multiple different habitats. And this could be a bias due to lower sampling of certain habitat types, but there could also be a signal there. So one way we can look at this is through the transition matrix. So this is when we track the changes along each branch of the tree to see what direction they might be changing their states. Are they going through this progression of becoming more and more specialists, like this model? But no, this isn't what the table shows us, and it actually looks something more like this. So although there are more lineages that go from no reducing habitats and expand into having the capability of going to reducing habitats, they're also likely and have shown a history of going into multiple habitats directly or straight into single reducing habitats. And there actually seems to be a stronger pattern of, once you're in a single habitat, remaining there or expanding into multiple reducing habitats. I think this actually makes some ecological sense in that it might be easier for a lineage or an organism to first move into one habitat type, and once it has the physiology or adaptations to live there or take advantage of those energy sources or develop a symbiosis, then it might be easier to expand and radiate into multiple habitats. So keep in mind that this is a generic level tree, and it's very rare to find one species that can occupy multiple habitats. So this is typically, this pink arrow here is probably representing diversification, but I still need to look a little bit more closely at each of the events. All right, so throughout this early exploration of gastropod patterns, which has finally culminated in our treatment of the patterns on a super tree, I became well acquainted with a particular group of charismatic limpets called the Lepetaloidea. And here they are here, aren't they cute? And they live in, but you can't really tell how special they are from this tree because their diversity is not really shown here, but they're really cool because they live beyond the vent seeps, whale falls, and wood falls, and have actually expanded into a wide range of organic habitats. And they can take advantage of even squid beaks, fish bones, crab carapaces, algal holdfasts, seagrass rhizomes, elasmobranch egg cases, and they have a huge range of different substrates that they've occupied, and these polychaete worm tubes as well. And so these are much smaller, they're very small substrates, and these are very small limpets. And so back in the 80s, Gerhard Haspernar was the main researcher of these limpets and had studied a lot of their anatomy and morphology and came up with this tree, which basically indicated that each family is specialized to a particular habitat type. But more recently, and molecular phylogeny revealed, as is pretty common these days, that this story is much more complex than that. And actually there are several different wood lineages, 3 different vent lineages, several different cold seep lineages, and some clades that live in a big mix of habitat types. So, given this information, I once had dreams of looking at the anatomy of all these different clades and testing it against the molecular phylogeny and figuring out, well, what's going on with convergence, and I had a lot of questions about that. But I ended up, it turns out this is very time-consuming work to do morphology on tiny limpets. So I ended up choosing to focus in on one particular clade to look at their detailed anatomy, and so I ended up working on the Lepetelidae. And so this is one of the only original families that remained monophyletic in the molecular tree. And this is the clade that lives on these sunken empty worm tubes. And so the family has a range from the Atlantic to the Mediterranean. And they live on these sunken worm tubes, and they have a unique radula, and they often leave scars on the worm tubes as well. And they can be found on the inside or the outside. And so I had an opportunity, as Dave mentioned, to go work with Gerhard Haspernar in Munich for 3 months. And while I was there, I built a 3D reconstruction of this particular species, Lepetella sierrai, which lives in the Mediterranean. And I also had a chance to go to Banyuls-sur-Mer with him after my time in Munich and try to collect fresh specimens. And unfortunately, we weren't able to get any, but you'll see I could learn a surprising amount from specimens that had been preserved and sectioned and stained 20 years prior to my arrival in Germany. So, given this set of slides with limpet sections on it, I photographed every other slide, so this on a 1 millimeter specimen, so tiny. And so we can't, with animals this small and of soft tissue, we can't do CT scans, if you saw Jesse's talk last week. And so the method we use is still is histology, which she also talked about, but in a slightly different way. And so once we have these stack of sections throughout the entire animal from front to back, we can align them in this program called Amira, and then go through the image stack and label each set of organs with a particular color. So once we have those sort of sets of organ tracings, the program can make 3D rendered images of each organ and then put it together as a whole package. And then this can be embedded as a 3D image in a PDF. So it's a little bit hard to wrap your head around this image, so I'm going to play a quick video, if I can find my mouse. Oop. So that was the front of the limpet with its cephalic tentacles on top. You can see that most of the body is occupied by this large, highly lobed midgut and is surrounded by a dense bacteriocyte network. Both of these are pretty unique to this family of limpets. It has a reduced stomach region and also these large esophageal folds. It also has a highly muscularized buccal structure. So this is these are cartilages that are near the mouth, which help move the radula forward. The radula is the scraping tongue that all snails have to graze on surfaces if they're grazers. And so this is part of the reconstruction, and it doesn't show the reproductive system, but they are simultaneous hermaphrodites, which is not uncommon for the superfamily. So this is just a recap of those main features that I showed you. And based on this anatomy and knowledge of their habitats, I drew some ecological hypotheses that they have these many modifications for feeding. So the first piece of evidence is this highly muscular buccal apparatus and a unique radula. So these are all features that are setting apart the whole family but are represented by this species. And so this indicates they're definitely actively grazing their substrates. And then additionally, they have these esophageal folds, this multi-lobed midgut and a simplified stomach, which indicate that they have much higher surface area and are probably having to do a lot more active digestion. And thus, I hypothesized that they're actually feeding on the polychaete tube. And I forgot to mention before that the tube is composed of a sugar phosphate polymer and is actually a lot more flexible than other polychaete tubes. So they have the potential to be deriving nutrition from it. And then the other piece of strange anatomy in this group is this bacteriocyte arrangement. So it's this highly dense yellow bacteriocytes in the mantle rim. So this is in the very edge of the body of the limpet with very thin epithelium covering it. And then this pink glandular tissue, which seems highly associated with the bacteriocytes. And we couldn't find any connections to the digestive system, and it seemed more oriented close to the external water. So I hypothesized that these microbes could be interacting with a reducing environment in the tube. If there's a strong lack of oxygen in the tube, there could also be sulfide there that they could be using. But an alternative hypothesis to chemosynthesis is that these microbes could be producing enzymes or other digestive agents to help break down the tube material. And so I said that it's not connected to the digestive system, but this isn't unheard of for mollusks. In fact, wood-boring bivalves, which are boring through wood and consuming wood, have symbionts in their gill, which then somehow send the enzymes to the digestive system to digest the wood. So although there's a disconnect here, that they could have that function. And unfortunately, since we couldn't get fresh material, we couldn't really fully determine what kind of bacteria these are. So that brings me into my last chapter, which is focusing on wood. And it's from the perspective of variation in the substrate structuring the diversity of the colonist community. And so this chapter resulted from a partnership with Jim Barry at Mbari, and I was fortunate to tag along on one of his research cruises to sink a bunch of wood and see what came back up on it. And since my question was variation in wood and how it affects the community, I tried to find as wide a variety of 10 different kinds of wood as I could. So although tree fern is not technically a wood, I had a chunk of tree fern. And I also had several different gymnosperm representatives, including ginkgo, torreya, stone pine, and redwood. And also a range of angiosperms, including Washingtonia palms, Dendromecon tree poppy, coast live oak, Calycanthus spicebush, and the Lionothamnus, which is the island ironwood. And I chose all of these groups primarily to get a range of phylogenetic representation and structural representation, but also because they're commonly found, if not native to the California coast. In fact, most of my samples and wood material came from the Tilden Botanical Garden, who focuses on California native plants, and they were happy to prune a bunch of trees for me. So once I had this material, I broke it into 3 replicates of each kind of wood, wrapped it in a 3mm mesh, weighed, measured, and tagged them, and drove them down to Moss Landing, which is here in the middle of Monterey Bay. And so then we set sail for a 6-day cruise, of which my project was just one of many, and made our way I think on like the 3rd day or so to the site Deadwood, which is at a depth of 3,200 meters. So once we were at the site, very early in the morning, we deployed the wood with a benthic elevator, which is this large frame which has floats and a beacon and weights, and it basically goes over the side of the ship and flows down to the bottom. And then using that beacon, the remotely operated vehicle, which does the bulk of the work on these cruises, goes down and follows it and is able to access the wood contained in the elevator and drop it off along the bottom. So then the challenging part was just to wait for 2 years and hope that I had an opportunity to go back and get it. And so 2 years later, this is what it looks like. This is a large chunk of coast live oak, and you can see there's lots of galatheid crabs hanging around on the outside and some smaller ones on the inside. There's a bacterial mat on the outside and some evidence of boring from bivalves. And there was a large snail up there, it's right there. And this is in the midst of a background fauna, including these rat-tailed fish, which were frequently swimming around, and hagfish, sea pens, a few different kinds of sea cucumbers, and I know there's a sea star back here somewhere. And this was a place where another wood bundle was picked up, and you can see it's kind of darkened, and we'll get a better look at that when the ROV comes and picks up this piece here. Yeah, so you can see when it goes for the grab. So we tried these ropes on, but the ROV pilots decided they would just grab the mesh anywhere they could. And but you can see this blackened sediment underneath, and that's indicative of a lack of oxygen. And so we call that anoxic sediment. And this is due to mostly the microbial action, breaking down the wood and using that as fuel. And also we can see this crinoid hanging off the end of the rope. And so they were not drawn here because they're wood specialists, obviously, they're filter feeders, but they can get a better vantage point for filter feeding when they're located a little bit further above the substrate. So Mbari specially designed these woodfall capture devices, which are kind of like butterfly nets with special locking and closing mechanisms to help get our logs up to the surface. And it's kind of it's an awkward fit because it's kind of a larger log, but to get them to the surface without losing very much material. And they designed them to have a series of ropes to pull and break zip ties so that the ROV arms can handle this operation. Yeah, okay, there it goes. All right, so once the ROV has done this, and so we had to go back and forth each time to get a new one and go to the next wood bundle. So this happened over the course of a few different dives in which other scientists were doing other projects. And so we'd bring it back to the benthic elevator and then get it on board, where we'd have to quickly process all the samples. And luckily I had my lab mate Rosemary there to help with all of this. And so for each bundle we would unwrap it and photograph it and then rinse all of the animals off it and preserve it in ethanol, and then preserve any chunks of wood that had animals in it in ethanol too, so that if I ever did want to do molecular work on them, I could, but I didn't. And so actually, no, I did on one. Yeah. Okay. So although there was a lot to process and just get things preserved, we couldn't look in detail at any patterns on the ship. But even just from some cursory looks, we could see some obvious patterns already. So for instance, this is the Torreya, or the U, which has, you can see it's fairly complex, I would say, and has lots of small needles and twigs, and we could see that there are already a lot of small animals living amongst this sort of nest of material. And although there weren't very many large pieces of wood, some bivalves managed to bore down these tiny twigs, but there weren't very many. On the other hand, the coast live oak remained fairly solid and was bored chock full of bivalves, but only on the first couple inches of either end, and they didn't go through the bark. But there were lots of limpets living on the outside of the bark. But besides that, there wasn't a whole lot of diversity living on the wood because it was mostly solid still. So once getting all of the wood back to Berkeley, I enlisted the help of several undergraduate researchers and who spent many hours extracting delicate bivalves from wood and making sure we had every animal out of the wood. So this resulted in bags of basically splinters of wood that are still in the lab because I haven't let go of them. And then many hours of then further sorting that preserved material into as fine as taxonomic groups as we could. And then beyond that, I enlisted help from different taxonomists through sharing photographs and specimens with them. And in all, we identified over 7,000 individuals to the family level and found over 46 taxa or morphospecies and several new species. And so those new species are going to continue to be described and worked on by the taxonomists that I contacted throughout this process. So one exciting one includes this boring bivalve. This is the one I got DNA out of, yay. And Janet Voigt at the Field Museum in Chicago is going to be describing it. And so this is just one of many new things. So to get back to the main question, though, of how does the variation in the wood actually structure communities, my key hypothesis was that the macroinvertebrate would show distinct features between different wood types, and that this would be reflected in different richness and relative abundances on different kinds of wood, and that in a multivariate analysis we would see distinct clumping of different wood types, and that wood type would be explained as a strong variable. And a third way that I looked at this was to test whether certain groups of animals had affinities with different kinds of wood. So just to kind of look at a basic plot of the data, this is plots of the sums of each of the 3 replicates for each wood type. And right off the bat, we can see that there are pretty striking differences between different types of wood. So palm being having the highest abundance of animals and being mostly dominated by Tanaids, which I'll show a photo of later. And then redwood and tree fern having really low abundances of animals. And then in between, we can see a lot of different proportions of colors representing the different taxonomic groups that I pulled out here. But and I plan to do some more detailed diversity analyses with this, but haven't quite gotten there yet. But just to illustrate, the tree fern had barely anything on it, and this is actually what other researchers have noted when they pull up tree ferns in trawls in tropical seas. And so this is kind of confirming there might be something going on with tree fern, whether they have secondary compounds that are preventing colonization or something like that, but there's not a lot known about what tree ferns have chemically. So redwood, on the other hand, we know has high tannins, and people use them for building on land because they're resistant to infections by herbivores, and so they're probably also resisting colonization in the deep sea. But they do have these bacterial mats on the ends, which was kind of interesting. And then to look at the overall community structure on the different wood types, I did a multivariate analysis, and this showed that there's approximately 70% of the variation is explained by wood type, and this has a highly significant p-value, so I was pretty excited. Oh yay, wood type matters, and the statistics support it. And then to display this, I did a constrained coordination analysis because you can't display more than 3 dimensions. And from this figure, I'm not going to go into it too much, but basically the tighter ellipses show that the replicates of that wood type were all much more similar to each other. So within like palm and tree poppy and yew and ironwood, they all kind of had the same community types represented there. However, these larger ellipses, especially on these like stretched ones, were all sort of the log-like material, so pine, ginkgo, and redwood. And this indicated that certain replicates had some kind of animals and other ones had different kinds of animals, so it might not be as have as tight of associations with those. And then the coast live oak was kind of off by itself, and it shows this association with the bivalves. So again, I should note that we also have the taxa displayed here, and these are all overlapping and you can't read them, but they're crustaceans and polychaetes and coiled gastropods, and then down here we have limpets and anemones, but I think this pattern is mostly driven by the limpets since we only had like 2 anemones. And all right, so the oak is clearly off by itself and has bivalves, and as I said before, it's because it pretty much only had bivalves living in it. And then the ginkgo also had this pale ellipse here, which is mostly in sort of the bivalve direction, but it also has this point down here closer to all of the crustaceans. And this is because one of the replicates was dominated by this boring isopod, which is a new genus, I now know, and it was really exciting and surprising to find that critter as the wood came up. All right, so the last way that I looked at these patterns was by doing an indicator species analysis to see, which basically tests for each taxonomic group, what is the strongest association to a certain wood type, and then whether that association is actually significant. So just to illustrate a couple of examples, the palm was kind of a leading winner for having more animal associations with it, including these amphipods. And the most common amphipod we had were these large Bathycerodosis amphipods, and they're also a new genus. And then, although not significant, the Galatheid crabs showed a strongest affinity to palm, and also several of the other isopods. So these are not the boring isopods, but other kinds of isopods. But as I mentioned before, Tanaids were by far the most abundant organism living on the palm, and they were just chock full of the grooves and kind of wrapped up in the sediment. So initially they weren't that apparent, but you could kind of see little white animals in the sediment, and once you pulled them out, we ended up having well, these are small jars, but they're small animals, so, so there's a lot of them. And all right, so another kind of wood that had several associations was the ironwood, or the Lionothamnus, and these also were not significant, the crinoid and the pycnogonids, but this is because there weren't that many, and they probably aren't actually specializing on the wood itself, but are utilizing it as a heterogeneous structure there in the deep sea. And this is the case too for the anemones, which showed Lionothamnus being the winner for it. However, there was a highly significant association of limpets with the ironwood, and I think this is truly reflected because there were such high abundances of limpets living on both the bark of the branches and on the leaves. Okay, so the major implications I drew from all of these patterns and looking at the data in a few different ways were that I think these patterns are mostly driven by some dimension of complexity. And although I didn't have a quantitative way of measuring complexity across these wood types because they differ from each other in several different ways, this is something that I think that I observed and I think is reflected in the statistics as well. And this makes ecological sense just because certain animals like the crustaceans and polychaetes kind of need pre-existing holes or cavities to live in, and other animals like the bivalves require maybe a larger surface or at least a volume to bore through for them to live in and grow and then reproduce. So on the other hand, substrates that start as complex might support a higher diversity of all of these more mobile animals, but they might be excluding the boring bivalves. So I mentioned before that Torreya did have a few bivalves, and palm actually had no bivalves living on it. So whereas the less complex substrates like coast live oak might actually become more complex through succession by the bivalves. So this study only had one snapshot 2 years after deployment, but other studies have found that over time there are more holes, and these holes become vacated by the bivalves either from being predated on or potentially dying naturally hopefully after they reproduce. And an additional role that these bivalves play is that they enrich the surrounding sediment, and by digesting the wood, and then their fecal pellets are pushed out of these wood holes and then into the sediment, it makes this energy that was locked up in the wood available to other organisms. So they actually are playing a larger role there. So to test these ideas about succession and different starting complexities, I think the ideal experiment would be to take a range of complex structures, all composed of the same kind of wood, and then follow them through time. And then this might give some more concrete answers to these questions. But that's something I couldn't get to in my dissertation. So to summarize what I was able to investigate in my PhD, I looked at broad patterns of diversification and specialization in a highly diverse group, the gastropods, and tested whether they're following similar trajectories, which they're clearly not. And then I was also learned a lot about limpet anatomy and how much you can learn from tiny preserved museum specimens and the importance of those collections. And yeah, that was great. And then also the wood, I learned a lot about how variation in habitat can structure communities, even on, and especially on short time scales, but there's a lot of potential that this heterogeneity and range of complexity is operating at larger time scales and larger biogeographic ranges as well. And a lot of other researchers have sort of explored this on a larger scale level. And I think this has implications for larger scale patterns as far as amount and diversity of wood that's entering the marine environment and how that might be influencing and shaping the deep sea communities that rely on it and the evolutionary opportunities that those organisms have had through time. So I feel fortunate to have been able to explore so many different avenues of research and learn so many methods and live in several different places and work with so many different people, all of whom I have to thank here. And I'd also particularly like to thank my committee and my advisor, Dave Lindberg, for his continual support and big picture perspective, which I feel has definitely rubbed off on me. And I'd also like to thank all of the museums and people who have given me specimens that have supported my work and exploration of different dissertation topics, and all of the people who helped me in the field and the undergrads who spent so many hours processing specimens, and a lot of people who helped me with statistics and IDing specimens both here in Berkeley and around the world. I'd also, of course, like to thank all the institutions, including UCMP and IB, that gave me financial support, and all of my friends, family, and VLSB colleagues who helped keep me sane over the last 6 years. Thank you.
Questions for Jenna?
Darko?
And in the sunken wood, do you see a particular developmental stage for the organisms that were living there, perhaps as a whole or in particular groups? I'm thinking about maybe finding only juveniles or only adults or only males or females.
Actually, it was interesting in that in several groups I saw a range of different stages. And for some of the crustaceans, there were males and females and gravid females, but mostly non-gravid animals. And then for the bivalves, there seemed to be mostly dominated by one species, but there were a few other kinds of species. And they did have a large size range, but I'm not sure whether that was a reflection of age or just that they had different rates of growth through different kinds of wood, but they tended to be of a similar size range in a particular log. Yeah. Jim?
So I was definitely thinking about how long that wood must last down there. You've alluded to other people studying that, but you didn't really mention how long you think the wood, like of the scale that you put down there, how long that wood lasts, and whether the species that are on it tend to really, if you think they're really specializing on particular species of wood, or if it's a very stochastic process, and if you watched it for 50 years that all these things would turn up on these pieces of wood.
Right, so that's kind of what I was trying to speak to in the end there, was that maybe over time, and we saw this in my colleague Craig McLean's work, who was also out on this cruise. He had wood that had been down for 5 and 7 years and was thoroughly bored through, and there weren't living clams on some pieces, but there were lots of higher diversity of polychaetes and snails living in there. So that's why I think there is sort of there's definitely a successional dynamic that other people have looked at, but I think the dynamic might change depending on like the starting complexity of the wood. And I don't think that there are strict specialists to certain wood types, but that certain structures might favor certain animal guilds, I guess. Yeah. Yeah, Jim?
Jenna, you mentioned secondary metabolites might be important for ferns and redwood. Do you know anything about what some of these other species have, and if there's any relationship between even the abundances or the species richness in phenolics or something like that?
Yeah, so I've tried to look up the secondary compounds and metabolites for all of the different wood types, and either I'm not using the right search terms or there's just not really a lot of information for the specific study, for the specific species that I looked at. And it seemed like actually most of them did have some sort of secondary compound or another, or is noted that that's toxic to cattle or something like that, but I didn't feel it was really concrete enough to draw those parallels. And I think just it would be better to do a more uniform study, but targeting, levels of toxicity or levels of complexity to see what roles those two things play. But I do think they're both important. Yeah, Jonathan?
Jenna, thanks for the nice talk. Did you see any patterns of like life history strategies amongst the diverse animals in the wood, where some had really large broods of embryos and some had smaller, larger embryos or anything like that on the different wood types?
So at this point, I haven't cataloged all of that data. It's been more just like noting like this jar has gravid animals and this one is not. And so it's not something that I've actually taken data on, but I think that would be an interesting question to see. Yeah, Carol?
So we're up to 8 bivalve families now that have bacteriocytes in the gills. Why do you think we're not seeing very much with respect to gastropods? Is there some basic difference there?
I have no idea. I think maybe for the gastropods, they might have more flexibility in acting as scavengers or predators, and actually, a lot of them seem to just be grazing the bacterial mats. So maybe who knows whether they're developmentally constrained to develop those bacteriocytes in the gill, whereas a few of them have done it clearly, so they have had the ability.
It's an infaunal, epifaunal difference.
Yeah, that could be a difference too of like having consistent access to sulfide. So a lot of the bivalves can access sulfide in the sediment using their foot. But so there might be just a few basic differences. But I don't know. Okay, Wayne?
If you didn't have to enclose them in bags and all the predators could get at them, how would it be? Would it be very different when you find the loft, or are there no predators, large predators?
Well, I guess I'm not sure. I think there were, the crabs were attracted and were probably grazing, but potentially being predators, and those large snails are predators. So there could be a difference if they weren't protected by the bags, but
Large fishes?
I don't know. Jim, what do you think? Do you think rat tails would target stuff living on whale falls?
Yeah, definitely.
Yeah?
It would have been nice to have different sizes of mesh and sort of control for that.
Yeah.
Would have been nice to have plastic simulators as well. That gets at the complexity issue a little bit too.
Yeah.
There's a lot of ways to do it, but that was a great start.
Yeah. So it's very preliminary, but.
Okay, I'd like to thank Jenna again, and please join us down the hall for cake and champagne.
Thank you.